In this unit, we're going to look at osmosis. And by the end of this particular unit, you should be able to describe the process of osmosis and also explain the effects of osmosis in living things. Okay, so let's dive in. Osmosis can be defined as a movement of water molecules from a region of high water potential to a region of lower water potential through a selectively permeable membrane. Okay, now let's try to analyze the definition of osmosis. Okay, water potential. What does water potential mean? Okay, what it simply means is measure of the capacity of water molecules to move from one solution to another. Okay, now molecules of water can diffuse freely across a membrane, but always down their concentration gradient. Okay, so water therefore can diffuse from a dilute solution to a concentrated solution. Okay, here is a diagram showing two solutions with different concentrations separated by a membrane, and a key showing water and solute molecules. If you try to analyze the diagram, in which direction will be the flow of water molecules? Will it be from A to B or B to A? Definitely the flow will be from B to A. Okay. Water molecules will diffuse from B to A, causing an uprise in volume of solution A and a decrease in solution B. When we're looking at osmotic effects, we basically focus on a cell placed in a solution. Okay, so there are three types of solutions an organism may be exposed to, and these include hypotonic solutions, isotonic solutions, and hypertonic solutions. Okay, so hypotonic solution is a solution of lower concentration than a cell. What this simply means is the solution has more water molecules compared to the amount of water present in a cell. Okay, so water can diffuse from the solution into the cell because the cell has lower water molecules compared to the amount of water which will be present in the solution. Okay, isotonic solution. Isotonic solution is just a solution of equal concentration to a cell. What this simply means is there is equal amount of water molecules in a cell and the solution. So water can freely diffuse in and out of the solution. Okay? Hypertonic. A solution of higher concentration than a cell. Now what this simply means is the solution has lower water molecules compared to the amount of water present in a cell. So water would diffuse from a cell into the solution. Okay? How are animal cells affected by osmosis? When an animal cell is placed into an hypotonic solution, it gains water by osmosis. As a result, it swells and eventually bursts. The bursting of an animal cell due to osmotic gain of water is called cell lysis. Okay? Now, when a cell undergoes this particular process, it is referred to as a lysed animal cell. On the other hand, when an animal cell is placed in an hypertonic solution, it loses water by osmosis. As a result, it shrinks. The shrinking and wrinkling of an animal cell due to osmotic loss of water is called cell crenation. Now, when an animal cell undergoes this process, it is referred to as a crenated animal cell. How is a plant cell affected by osmosis? Okay. If a plant cell is placed in an hypotonic solution, it gains water by osmosis. As a result, the protoplasm increases in size and starts pressing against the cell wall. This process is referred to as cell turgidity. Now, a plant cell that has undergone this process is referred to as a turgid plant cell. Okay? On the other hand, if a plant cell is placed in an hypertonic solution, it loses water by osmosis. As a result, the protoplasm decreases in size and starts pulling away from the cell wall. Okay? This process is called plasmolysis. Now, a cell that has undergone this particular process is referred to as a plasmolyzed plant cell.